This podcast comes to you from the PlasticHeal project, exploring micro and nanoplastics effects on human health. We will be talking today with Thierry Rabilloud, a researcher from the French National Center for Scientific Research. He is one of the authors of the scientific paper Does Size Matter? A proteomics-informed comparison of the effects of polystyrene beads of different sizes on macrophages. The aim of this study, published last summer, was to get a better appraisal of the effects of plastic particles on macrophages, which are important players in the immune system. Is it right?
Yeah, the idea that behind this paper is that we don't really know the influence of size on the effects that nanoparticles or microparticles might have on cells. There is a general claim that says that the smaller, the worse, because it is believed, and it's true to an extent, that smaller particles can penetrate more cell types and may have effects on some cell types that the larger particle won't be able to penetrate, and they will have no effect. So we chose macrophages for a good reason, that is that this cell type is the scavenger cell type for everything that is a particle in the body. So they are what we call professional phagocytes. Their profession is to eat. Everything that is particle can be bacteria, plastics, lipids, silica, dust, whatever. As soon as it is a particle, that's for them. The point is that when they eat a particle, that's not inert for them. In a sense, they taste it. They try to understand the nature of the particle, if it's dangerous or not. And to do that, they have receptors that bind molecules. And normally, in the wild, they are molecules oriented more towards bacteria or viruses components. So if they detect something that tastes ugly, for example, a bacterium, they will start an inflammatory reaction. If they taste something that tastes good, and quite surprisingly, cell debris tastes good, they will start an anti-inflammatory reaction and promote tissue repair. So the question is, what do plastic particles do to them? And are the extent of the effects and the effects themselves depending on the size? Because macrophages, they are completely able to eat anything from 5 nanometers to 10 micrometers. And that's not a problem for them. And if it's bigger than that, then they will cooperate to try to englobe the nasty big particle, and that's called a granuloma. Okay. And that's well known. So we know that they are able to handle every type of particle. We know that this is their function. And we know that they can start inflammatory reactions that can be deleterious. For example, in the case of silicosis or asbestosis, which are chronic diseases, that's the permanent activation, inflammatory activation of macrophages that builds the disease. Okay. So it's very important to know if plastics, for example, are able to start this type of inflammatory reaction. And, for example, is it size dependent or not? That we felt we have to know. Above that, the core idea in this paper was to use a wide scope approach. Instead of asking a series of questions where we are bound by existing tools and our imagination, in a sense, we used an omic technique, which basically says I try to determine what changes between a control cell that has never seen any particle and a cell treated with a particle, and several particles. And everything that will change is interesting. And in proteomics, what we examine as an object is proteins. Okay, so we make this screening process at the protein level. Okay, so the basic idea was to take macrophages that were murine macrophage cell line, which are easy to grow and are well responsive, and treat them with particles, fluorescent particles of polystyrene, which are available of different sizes, every time at the same concentration, which is known to be non-toxic. Okay? Because we don't want to see dead cells. The response of dead cells is not very interesting. So they are still alive, and they do respond to this concentration. Okay? And we see that at the proteomic level, and we compare the responses at 3 different sizes that were selected, each 1 order of magnitude bigger than the other. So we compared the different responses. There were many in common, which is rather reassuring. That means that the size has an influence, but not that much. The nature of the particle has a bigger influence than that. So we know from previous experience on another model that study that the response that we see is specific for polystyrene. But even though most of it is size independent, there is some size dependency on the responses. And in fact, the bigger, the worse.
It was the opposite that
exactly
was assumed that.
But because that's probably because the macrophages are able to handle big particles. Okay, they have ingested the particles, they are not building an inflammatory reaction that we have checked. But that does not mean that there is no effect. Okay, there is an adaptation, and that means that something has to be done to accommodate the particles. If by chance in our lifestyle we have something else that exactly acts on the same proteins, maybe it will become too much and the cell won't be able to cope with it. Okay, but that we can't predict because we are so different in our professions, lifestyles, etc. And we are different, each one from each other. The idea of doing proteomics is once we have documented what changes, and if we know that for many, many different substances, we can predict the couples that will interact each one with each other. For example, something that we have seen too is that when a macrophage has internalized particles, then it becomes less responsive to bacteria, which is not good because they are there to be responsive to bacteria.
Of course.
Okay?
See, they are not
that
like being harmed by the particles, but they are using an effort, they are making an effort to work.
Exactly. But then when you come with something else afterwards, they are less efficient. They are not completely depressed. They are just a little bit less efficient. The point that arises next is that we have done that for 24 hours. So we have given a given dose. But we don't know how long the particle persists in the macrophages. And we don't know if they will accumulate during the lifetime. So in a human lifetime, we don't know if these accumulation problems will exist because we don't know how, for the time being, we don't know how long a plastic particle persists in macrophage. Because the macrophage will try to degrade it. They are very good at that game. But there are some things that they don't degrade. And for the plastics, we don't know yet. We have performed, after the paper, some experiments that suggest that at least for 2 weeks, they don't degrade. So probably the plastic stays for long. And if it stays for long, then comes the problem of what happens after decades of ingesting nanoparticles and nanoplastics. Not 1 day or 2 days, decades. Unless we have performed the experiment, we don't know. That's coming. That's up and coming.
And I look forward to hearing soon about the new conclusions that this experiment may offer. That's it for this episode of the PlasticHeal podcast. Many thanks to Professor Thierry Rabilloud. The PlasticHeal project has received funding from the European Union's Horizon 2020 Research and Innovation Programme. For further information about the project and to find out much more about the results and findings of PlasticHeal research, please visit our website at www.plasticheal.eu. You will also find us on social networks at Instagram, Twitter, and LinkedIn.